Today, our speaker is Christina Annunziata. She got her MD degree at Georgetown University Medical School. Subsequently, she joined NCI and worked in the lab of Lou Stout. And then she worked with Elise Cohn. And now she's in the women's malignancy branch. She's head of the translational genomics section. Her title, Ovarian Cancer in the Genomic Era. Tina.
Okay, thank you very much. Okay, so I was actually just looking back at my previous presentations, and I realized this is the 8th year that I'm giving this lecture, but I wasn't always called the genomics era. So this is definitely for 13 years. I know. So this is definitely updated because it wasn't previously genomics. Anyway, so thank you for inviting me back to speak again. I wanted to focus on the topic of genomics and functional genomics to start out with, and I'll get to ovarian cancer in a little bit. So what is genomics? What is cancer genomics? I think genomics, in my view, is the study of the whole genome. So not individual genes or individual mutations, but study of the genome in general. Either chromosomes, gene expression, but some sort of global analysis. So I would say the genomics era actually started back in 1959 with Knoll and Hungerford, who studied chromosomes, and they identified a recurrent abnormality that they called the Philadelphia chromosome, because they were in Philadelphia, that was occurring in patients with this chronic form of leukemia, actually. And this is what it looked like. So in 1959, they looked at the chromosomes from multiple patients with this chronic form of leukemia, and they noticed that everybody had, or most of the patients, had this little thing here with the arrow. Where's my thingy? I don't know where it is. There. A little extra chromosome. It looked like an extra chromosome. So they studied this. Eventually, in 1973, Janet Rowley discovered that this little chromosome was actually a reciprocal translocation between chromosomes 9 and 22 in all of these patients with, which was at that point called chronic myelogenous leukemia, or CML. And then in 1984, Goff et al. Discovered that this little chromosome was a recurrent fusion between chromosome 22 and actually 19, but here he found that it was a recurrent fusion between BCR and ABL. So the gene BCR, which is he called breakpoint cluster region, and ABL, which is a kinase. And then it wasn't until 1996 that we actually came up with a drug called imatinib, which here it's called CGP 57148, with a drug that actually blocks the ABL kinase function. So that was quite a time between 1959 and 1996 before we came up with therapy for this particular thing. So that points us to what is actually functional. So the therapies we use are actually blocking a function of something. So how can we use functional genomics, not just observational genomics like they did in 1959? Can we use sort of a functional genomic experiment to find out what part of the genome is actually doing something, what is causing an effect, and what can actually transform normal cells into cancer, these so-called driver alterations. So in 1981, I think she et al. Performed a first functional genomic experiment. And this is what led to the discovery of what we now call HER2/neu. So in this paper in 1981, these researchers published that they had found transforming genes of carcinoma and neuroblastoma into mouse fibroblasts. So what they did was they took the entire DNA of neuroblastoma, that's why it was called neu, neuroblastoma, chopped it up, put it into non-transformed fibroblasts, and saw which part of the genome actually caused an effect, which was to transform the cells into cancer cells that could actually form tumors in mice. So they found a gene that they had called NU, and then in 1984, Schechter et al. Noticed that the NU gene was actually quite related to the EGFR, the epidermal growth factor receptor. This is by a southern blot. And then in 1985, Cousins et al. Found that this area of transformation was on chromosome 17, and it was actually amplified. So you can see in that little blob on the side that means that there's like a ton of it. So it's a lot of it. So it's amplified in this region of chromosome 17. And then, of course, in 1987, Dennis Slayman et al. Discovered that it was recurrently amplified in a fraction of breast cancer cases. So this is now a southern blot of chromosome 17 in all of these, or this region of chromosome 17 in all of these breast cancer cases, and you can see that in some of them it is extremely amplified. And that eventually led to, the development of an antibody to block this receptor, and then the approval of Herceptin or trastuzumab, which is a drug that we use today in breast cancer that are known to have HER2 amplification. So I would say this is probably the first functional genomic experiment. So what about ovarian cancer? Can we use genomics to study ovarian cancer? And do we have any drivers in ovarian cancer? So now I will introduce ovarian cancer. So getting to the actual subject of this particular TRACO lecture, ovarian cancer, as you may know, is the most lethal gynecologic malignancies in the United States. It causes greater than 16,000 deaths per year, and it's the fifth most common cause of death in women. And this is because 70% of women are diagnosed with advanced ovarian cancer. It's unusual to find ovarian cancer in early stages. Less than 35% of the advanced stage patients are alive at 5 years. And this is what we mean by advanced stage. So stage 1 is early stage, and that occurs in about 20% of cases. But the survival is quite good. The survival is 90%. If you can find stage 1 confined to an ovary, your survival is quite good. Stage 2 has left the ovary but is still in the pelvic region. Stage 3 has spread intraperitoneally or to lymph nodes, and that, like I said, is the most common stage to find ovarian cancer diagnosed in. Unfortunately, the survival drops to 45% at 5 years. And then stage 4, the survival is even worse at less than 5% at 5 years. So what is the treatment for newly diagnosed ovarian cancer? Of course, the first step is to complete surgical staging, and I'm going to show a little bit about that. Optimal reductive surgery, which we'll talk about, followed by chemotherapy, and then, of course, clinical trials. So complete surgical staging, did I take that out? I think I did. Sorry, I took out complete surgery. Complete surgical staging involves looking through the entire peritoneum and looking for lymph nodes. And as you can imagine, the pelvis and the abdomen are sort of contiguous. So the difference between stage 2 and stage 3 is actually just an anatomical distribution and not a full hard line of separation. And the peritoneal fluid circulates freely throughout the abdomen. So the surgical staging needs to include sampling of the diaphragm, of the liver, and of sort of everywhere in the abdomen, including some of the retroperitoneal lymph nodes, because of the way the peritoneal fluid spreads. Optimal cytoreductive surgery means that the surgeon has left less than 1 centimeter of disease in the abdomen. That is the goal, and now the goal is actually to leave as little as possible, because as you can imagine, the survival of ovarian cancer improves with the less and less disease left at the time of surgery. So the best survival of ovarian cancer patients is if they can get to zero disease visible in the abdomen. Chemotherapy, the basis of chemotherapy is platinum in combination with a taxane, and intraperitoneal if the disease is stage 3, and optimal, meaning less than 1 centimeter, left at the time of surgery. So the reason for the combination of platinum and taxane is that you can see here the introduction of various chemotherapies and 5-year survival on the bottom of advanced stage disease. This is now stage 3 and stage 4 only. With the introduction, at the time of the introduction of alkylating chemotherapy in 1960s, there was basically no one who lived beyond 5 years if they were diagnosed with stage 3 or stage 4 ovarian cancer. When platinum was introduced in the 1970s, that was the first sort of big revelation, big jump up to 5% of people alive at 5 years in the 70s. So then in the 80s, people decided, well, we should combine them, alkylators, with platinum, and actually that was better, got up to 15%, 5-year survival in the 1980s. And then in 1990s was the introduction of the taxanes. So this is paclitaxel or docetaxel. In the 1990s, another big jump with the combination of paclitaxel and carboplatin, up to 35%. And then in the 2000s, there was actually not new chemotherapy agents, but a new way of introducing the chemotherapy agents. So instead of giving them IV, the thought was to give it intraperitoneal because that's where the bulk of the tumors are. And so when they did that, the survival went up to 40% in the 2000s. So this is survival at 5 years. But from a molecular standpoint, I mean, I think we still don't know very much about ovarian cancer. It is a very heterogeneous disease, and it's heterogeneous even at the point of just looking at it. So if you think of ovarian cancer histology, ovarian cancer is made up of at least 5 distinct histologic subtypes. The most common, as I have here, is the serous. So high-grade serous ovarian cancer is the most common and also the most the worst prognosis, and that is a particular type of ovarian cancer, which is shown in A. And then you have low-grade serous, which is more papillary, which is shown in B. And then in C, you have endometrioid cancer, and D, you have a mucinous cancer, and then in E, you have the clear cell cancers. So you have at least 5 distinct histologic subtypes. So if they look this differently on the microscope, you would think that they are probably very distinct, at least at a molecular level as well, right? So, I mean, we're treating them all exactly the same way, but maybe we could improve if we figure out what is actually different between these cancers. The tissue of origin is also potentially different between these cancers, and some recent work has shown that perhaps serous cancers arise from the fallopian tube and not the ovary itself. Endometriosis could potentially give rise to either the endometrioid subtype or the clear cell subtype. And then the Müllerian epithelium or extrauterine epithelium might give rise to mucinous or other types of ovarian cancer. So very heterogeneous, even from the point of a cell of origin. So the hypothesis is that if we increase our understanding about biological and biochemical events underlying ovarian cancer progression, we can create avenues for new treatments. So maybe we shouldn't be treating them all the same, or maybe we should give them all the same chemo, but maybe give them an additional agent specific to their mutational status. Can we use genomics to figure this out? So I'm going to start with a sort of an overview of each type of cancer and show you where we are so far. I could tell you the punchline is that we don't have specific therapies as of yet, but we're probably getting there. So let's start with clear cell or endometrioid cancers, which seem to be related from at least a cell of origin standpoint. Clear cell cancers are 5 to 10%, as I mentioned, whereas serous is more common, 70%. Clear cell cancers actually do have a worse response to standard chemotherapy with the platinum and the taxane. And clear cells are associated with endometriosis in up to 40% of cases. So this study back in 2010 looked at what are the genomics of clear cell ovarian cancer. So they started out by sequencing RNA from 18 cases of clear cell ovarian cancer in one cell line in a discovery phase. Then they went on and they sequenced exons only from 210 additional samples, 101 more clear cell, 33 endometrioid, 76 serous as sort of their negative comparison, and another clear cell line. And then they immunostained, they did immunostaining for interesting things in another 455 samples. So a pretty big study. And what they found was that they found ARID1A this is a gene that has recurrent mutations in clear cell and also in endometrioid ovarian cancer. So that still didn't come up. So this was basically just supposed to show you a picture of ARID1A mutations across. They actually they span the entire gene, so they're not in one recurrent location, but it's the gene itself. Anywhere in the gene is mutated. So it's sort of it's not a particular kinase location, for example. It's anywhere in the gene that can be mutated. So what is ARID1A? And this kind of gives us a clue as to why it's not a specific location. So ARID1A is actually in a chromatin remodeling complex. It has found to be mutated in breast cancer and lung cancer previous to this publication in 2010. It occurs in 1p, on chromosome 1p36, and it's actually deleted in 6% of all cancers across the board. So this suggests, and the mutational spectrum across the gene itself suggests that it's probably a tumor suppressor gene. So it's not an activating oncogene. It's probably a loss of function mutation that's happening. It's a loss of function of the ARID1A gene. So they did confirm that ARID1A expression was lost, and here on the y-axis it says loss of BAF250A, and that's the protein name for the ARID1A gene. So the protein was lost in 73% of clear cell cancers, and with the ARID1A mutation, and it was lost in 50% of endometrioid ovarian cancer cases that were associated with ARID1A mutation, but 0 high-grade serous cancers. So this suggests, okay, it is something that's maybe particular to clear cell or endometrioid subtypes, definitely not in serous cancers. So, to summarize, ARID1A was mutated or lost in over 40% of clear cell cancers, 30% of endometrioid cancers, these are ovarian cancers, and less than 1% of serous cancers. However, since it's a tumor suppressor, we're not really sure of what to do with this information. So we don't have an indication of which pathway is affected. There's unclear sort of a therapeutic direction here. What do you do with the loss of a tumor suppressor? It's a little bit difficult to target that in a way. But at least we are learning that there are specific mutations here. So we have potentially diagnostic utility, but I would argue that this is an interesting finding, but it didn't give us a therapeutic direction because it was not identified with a functional experiment. So there was nothing where they put in a gene, put in something from the genome and found an effect. No, we're just sequencing and we're making an observational experiment. So we're left without a functional outcome. Okay, so what about mucinous cancers? Mucinous cancers, we've known since 2004 or beyond that mucinous cancers have a worse overall survival compared to non-mucinous cancers. And this is just a survival plot of people surviving years since diagnosis of mucinous versus non-mucinous ovarian cancers. And then in 2006, this is kind of an overview of a gene expression experiment that looked at serous low-grade cancers, serous high-grade, and mucinous cancers, either grade 1 or grade 2. And they discovered that mucinous cancers, surprisingly, were, or not surprisingly, were different. From a gene expression standpoint, this was a very early gene expression array. I think they only had like 10,000 genes on it. It was one of the early ones in 2006. But still, even with this limited information, you could see that the mucinous cancers have a completely different gene expression profile than the serous cancers. So, okay, that is good. We found that, they found that perhaps KRAS was more commonly mutated in mucinous cancers compared to serous cancers. Again, this is an extremely small study. And similarly, in low-grade serous cancers, which are distinct from high-grade serous cancers again, KRAS and BRAF were found to be mutated. So BRAF was studied at codon 599 and KRAS in codons 12 or 13, which are the most common sites of mutation, and was found, interestingly, that 68% in this small study of low-grade serous cancers and 61 of serous borderline tumors, which are kind of a precursor to low-grade serous cancers, had one of these two mutations, but none of the high-grade serous cancers that they studied had this mutation. So another clue that is pointing us towards either mucinous or low-grade serous cancers, perhaps we can target KRAS, or the RAS pathway. This is just a graphical representation of what I just said, is that the serous borderline tumors and the low-grade serous cancers were more commonly mutated than the high-grade serous cancers, endometrioid or clear cell. There you go. So how can we target RAS signaling? I think probably everybody has heard of the RAS pathway because we have this huge RAS initiative at the NCI. But how can we target it? One way is to target downstream of RAS, and that is at the point of, oops, do I have this? Here, at the point of MEK. So RAS signals to RAF, then to MEK, and then to ERK, which then goes in as a transcription factor and changes gene expression. So Farley et al. Decided that perhaps we should try the MEK inhibitor called selumetinib in women with low-grade serous ovarian cancer. So they treated 52 patients. They saw 8 responses and 34 patients with stable disease greater than 4 months. So that sounds promising, but if you look then at the mutational status of these women, the punchline here is that there were responses in 5 patients who had no BRAF or KRAS mutation, and only 2 responses in 16 patients who had a mutation. So actually, the responses were more common in people without the mutation than with the mutation. So that didn't quite go as planned. So where do we go from here? So as I showed you before, the RAS pathway was neatly described as going from RAS to RAF to MEK to ERK. However, that is probably not the case in cancers or in other cells as well. RAS signals through many other pathways. So if you have a RAS mutation, the cell probably doesn't care too much about MEK because it has a lot of ways of getting around that. So that's probably why it didn't work. So it was not a driver. So again, we found this mutation by sequencing, but we did not find the mutation by doing a functional experiment. We just said, wow, there's a KRAS mutation, but there were no functional experiments to say that the RAS was actually a driver. And even if it was a driver, MEK is not the driver. That's where we are with that. Okay, so what about high-grade serous ovarian cancer? So this is the most common type of ovarian cancer. As I mentioned, 70% of cases are diagnosed as high-grade serous. Okay, good, that worked. So high-grade serous cancers were studied back in 2011, published for the first time as part of the Cancer Genome Atlas. So I think it was like the second or third cancer that was presented in the Cancer Genome Atlas. Included clinically annotated high-grade serous ovarian cancer samples, identified molecular abnormalities that influence, well, the goal was to identify molecular abnormalities that influence pathophysiology, affected outcome, and constitute therapeutic targets, potential therapeutic targets. So what they did was another, what I would call observational study with 489 high-grade serous ovarian cancers. They looked at messenger RNA expression, microRNA expression, DNA copy number, promoter methylation, and they did whole exome sequencing on 316 of those samples, so a subset. So the sample inclusion, of course, they had to be newly diagnosed patients, high-grade serous ovarian cancer, no prior treatment, and they had to have a companion normal tissue, either adjacent normal tissue or peripheral lymphocytes or previously extracted germline DNA. So that was for the purpose of this experiment, which was the copy number analysis. So what this is showing you is on the y-axis, so to speak, are the chromosomes, so chromosomes 1 through 22, and on the x-axis is individual cases. So these are 489 cases, and they're showing you here high-grade serous ovarian cancer compared to the previously studied TCGA samples of glioblastoma multiforme. That's what GBM is. So what you can see here, well, and then the scale is that red is amplified and blue is deleted. So what you can see here is that in glioblastoma, there was a relatively quiet genome, and there were distinct areas of either amplification or deletion. But in high-grade serous ovarian cancer, the amplifications and deletions are kind of all over the place. So you see kind of distinct areas of recurrent amplification or deletion, but they're like all over the genome. They're in all the chromosomes. There's some distinct ones, but it's really not pointing to it. So the conclusion from this was that there is a high amount of genomic disarray or chaos in ovarian cancer that is not seen in other cancers. So is this a problem with DNA repair? That's leading us towards something interesting. Significantly mutated genes, P53, of course, was the most commonly mutated. BRCA1 or 2 was the next most common. BRCA1 is on the top and BRCA2 is on the bottom. If you combine them together in the same pathway, that's the next most common pathway. And then a number of other ones. But as you can see, nothing really sort of jumping out at you as anything besides P53 and BRCA. There, sorry. I should have pressed the button first. Okay, so if you focus there on this idea of genomic disarray and homologous recombination, which is a way of fixing the genome, we know that BRCA is altered in 33% of cases, either by deletion, mutation, or methylation. So if you look at the survival, actually the BRCA mutated cases actually do better, interestingly. So the BRCA mutated here on the left is in the red, and the methylated and wild type is in the black. So the thought was that with all this genomic disarray and the recurrent BRCA mutations, maybe there is a common theme in ovarian cancer as defects in DNA repair. And so they looked at, well, where else do we have mutations that we've seen? Where else do we have alterations? And we could say that maybe in 51% of cases, not just the ones with the BRCA mutation, in 51 overall, we maybe can find a specific defect in homologous recombination or DNA repair. And then this is sort of another way of putting it all together. This is looking at 84% of cases have either DNA repair or cell cycle problems in FOXM1 signaling. So we have DNA repair and cell cycle. Maybe that accounts for ovarian cancer. Who knows? So what next? So what can we do with this information? It was not a functional experiment, but maybe we came up with potentially functional therapeutic targets with respect to homologous recombination defects. And one of those drugs is PARP inhibitors, which I'm going to talk about in a little bit. They also found commonly deregulated pathways such as RB and RAS, PI3 kinase, and they also found aberrant genes or networks. So they're saying now we shouldn't look at just one individual gene, we should look at networks of genes that might all be affecting the same function. Okay, so targeting homologous recombination DNA repair, potentially with PARP inhibitors. Just to back up, the BRCA mutations were originally found, published, I guess, for the first time in 1990 by Hall and King et al., who discovered that there was a particular part of the genome that was altered in families where there was this recurring breast cancer throughout. So if you look at this pedigree, you could see that the solid, the circles are females and the squares are males, and then the solid are the ones that are affected with the cancer. So you can see that there's a recurring hereditary breast cancer in each of these families, which affects about half of the women. So these people identified eventually the BRCA gene as or chromosomal locus at that point, the BRCA gene that was recurrently mutated in these families. So in high-grade serous cancer, the BRCA, like I mentioned, was about 10 or 15% the BRCA mutation in the germline, but then also BRCA can be mutated in the cancer itself and not mutated in the peripheral DNA, in the genome. It could also be methylated, but then other things in the DNA repair pathway, such as the EMCY amplification, PTEN loss, or other homologous recombination defect. And this is where we think that in about half of the cases, we can find genetic evidence for defects in homologous recombination. Interestingly, more recently, people have looked at what are some genes. So instead of just looking at copy number alterations or methylations, can we come up with a gene signature of homologous recombination defect? So these people looked at cases with BRCA mutated BRIT1 or RAD51 and found that there were 230 common genes that might present a signature of homologous recombination deficiency. And this is actually looking in breast cancer. Patients with homologous recombination deficient, again, had a better survival than homologous recombination intact. And if you look in ovarian cancer cell lines, even the case of two different cell lines, one with homologous recombination intact signature and one with homologous recombination deficient, the actually deficient one is more sensitive to the PARP inhibitors. So what are PARP inhibitors? PARP inhibitors are inhibitors of single-strand DNA repair. And this is a specific cartoon of how we think it works in BRCA mutated cancers. So in the normal situation, you have your DNA suffers single-strand breaks by various processes, such as cellular metabolism, exposure to the environment, etc. So what happens is that the PARP enzyme comes in and marks these single-strand breaks for repair. So if you block the PARP in replicating cells, the DNA, the single-strand break, gets converted when you untwist the DNA, gets converted to a double-strand break. So now you have a double-strand break, which is dependent on homologous recombination or repair that uses the BRCA1 or the BRCA2 gene in this pathway. If you have cells that are deficient in BRCA, they cannot repair this, and they get sort of overwhelmed because they don't have a homologous recombination pathway intact. They have huge amounts of double-strand breaks because of the PARP inhibitor, and they would selectively die. That is the hypothesis. So a PARP inhibitor that we've done multiple clinical trials with is olaparib, or AZD2281. This is an orally available PARP inhibitor, and it has been shown to have synthetic lethality in BRCA mutant cell lines. And interestingly, it was just approved for use in third-line treatment of women with BRCA mutation and recurrent ovarian cancer. Our study was to take it further, so going from single agent to combination with chemotherapy. And the idea here is that the carboplatin is going to induce a lot more single-strand breaks than just environmental exposures and cellular metabolism. So we're going to give people carboplatin so that we can cause single-strand DNA breaks, and then we're going to give them the PARP inhibitor. And if they don't have an intact homologous recombination, either because of a BRCA mutation or because of one of the other, 35% of women who don't have a BRCA mutation that have evidence of defects in homologous recombination. So cohort 1 was BRCA mutant, either breast or ovarian cancer, and that was recently published in JNCI 2014. Cohort 2 was triple-negative breast cancer, which may, in recent TCAGA analyses, have similarities in this homologous recombination deficiency, similar to what's found in ovarian cancer, non-BRCA mutant. And then cohort 3 is high-grade serous ovarian cancer, again, with the thought that even with a normal BRCA, there's still a 35% chance that you might have a defect in homologous recombination. And these were both presented by Victoria Chu in our group at AACR 2014 and then recently at ASCO this past year. So what we found was that the safest, we were able to dose escalate, and we found a safe dose of olaparib, 400 milligrams twice daily, and carboplatin AUC5 in the mutation carriers, which was comparable to what we found in the non-mutation carriers, either breast or ovarian cancer. So as I mentioned, this phase 1b study was recently published in 2014. We enrolled 45 patients, 37 ovarian and 8 breast cancer patients. This is only in the BRCA mutation carrier cohort. Dose escalation in additional 30 patients, dose escalation in 30 patients and phase 1b expansion at the maximum tolerated dose in additional 15 patients. And happily, we found that we had hit on something quite interesting. So this is patient, the colors are the dose levels, but you can see at all dose levels, we have people responding to this drug, even though they had previously not responded to platinum alone. So with the addition of the olaparib to the platinum, we are getting good responses. We're getting deep responses. We've had 3 patients that have been on study for more than 5 years that have on this particular cohort. So, so this was extremely promising. This is just to summarize that we had, this says no complete responses, but since then we've had the complete responses. We had in the ovarian cancer cohort a partial response rate of 44%, stable disease of 38%. So an overall clinical benefit rate of 82% in these women with previously chemo-refractory disease who have been given the carboplatin in addition with the olaparib addition. So this is very promising. So now we think we've hit on actually something functional that we can work with. So the conclusions were from that trial that it's well tolerated in combination with carboplatin, highly active in advanced chemo-refractory BRCA deficient cancers, more activity, of course, at the higher dose, but a positive proof of concept that we have activity and tolerability in this genetically defined targeted therapy with BRCA mutated cancers. We did present the results of the high-grade serous cancer at the ASCO 2015 meeting. I don't have the results here, but those were very promising as well. So that brings us to exploration of new targets. So how can we now go from using the observational sort of gene expression mutational analysis to going towards a more functional experiment? How can we sort of recapitulate what was done back in, 1981 with the HER2/neu? Let's just step back and say there are a lot of tests that are being marketed out there for these, quote, actionable mutations. But when you hear this, this is not things that have been functionally validated. These are things that might potentially be targets of therapies that exist. and you could see them being sold by some places like Caris or Foundation One, and you get back a report that you have this KRAS mutation, for example, that is possibly sensitive to drug XYZ, or you have something that is unlikely to benefit from drug ABC. So you're getting these reports that I think are more observational than functional. What we have ongoing right now in the NCI and also around the country are basket clinical trials to test these hypotheses that if you have this mutation, are you actually sensitive to drug X, Y, or Z? So what these people are doing is that they're going to sequence the tumors and then assign treatment based on a mutation. These are hypotheses that are being tested. These are not definitive going to respond. Typically, as I mentioned, there's no functional link other than, we introduced this mutation into a cell line and half of them had a response. So what are actionable mutations? This is a very interesting editorial that by Redig and Yanni that was just recently published in JCO, which I thought this was quite apropos. So they say actionable mutations, they're talking about these basket trials, actionable mutations depends in large part on the strength of the data linking the target and the targeted therapy. For this trial designed to work, 2 key conditions must be met. Number 1, the tumor must depend on the target pathway. And number 2, the target therapy must reliably inhibit the target. So you see, we have a lot of information missing. We do need to do these trials to figure out what works in what situation, but a lot of the information is still missing, which I think these 2 key concepts are sort of a hurdle, what needs to be addressed in this whole push for personalized medicine. Achieving both goals can be a matter of some complexity. Indeed. Okay, so how do we get actionable targets? From an experimental standpoint, I think we do need a functional experiment. So how can we use functional genomics? So one way that can be done, published in 2006 by Dr. Lou Stout at this institution, is a functional genomic approach using an RNA library, an shRNA library. So what they did was they had a library containing shRNAs, between 6 and 12 shRNAs targeting 2,500 different human genes. They say they have 3 constructs per gene, but actually since then I know that they've updated that to 6 to 12 per gene. So there's about 15,000 constructs in this library, all sequence verified, and they contain a 60-mer barcode so they can identify which target sequences have been lost from the pool. The library contains protein kinases, PI3 kinases, deubiquitinating enzymes, NF-kappa B pathway regulators, and other genes of interest. So what this library screen does is that it gets infected into the cancer cells, and it's inducible. So you can induce it in half your population, and you can leave it off in the other half. And then you look for genes that are, or barcodes that are lost in your induced population and present in your non-induced. So you're really looking for things that are specific to this cancer cell growth and survival in culture. So we did this library in 4 ovarian cancer cell lines. We looked at 2 serous and 2 non-serous ovarian cancers. And we found that there were a number of genes, 55 genes that were common to at least 3 or 4 of these cell lines. Could these potentially be drivers? I don't know. So we have a list of genes that are common to serous cancers. We also have a list of genes that are common to non-serous cancers. And we are following up on each of these to see if we have actually identified a functional target. Like I said, we're following up in 6 additional cell lines, 2 different RNAi constructs, and we're selecting the targets that do have drugs designed for these genes. We are doing, in addition, we're doing focused functional screens in specifically serous ovarian cancers, and we've focused on the NF-kappa B signaling pathway as an example of what might be a targetable pathway. So if you go back to the Cancer Genome Atlas, one of the other things that they looked at was by gene expression, can we identify subgroups of ovarian cancer that look like they have something in common? Subgroups of high-grade serous ovarian cancer that have sort of gene expression patterns that are common. And one of the things that we became interested in was this immunoreactive subgroup. So it looked like there were a lot of genes that were potentially involved in immune inflammation or immune regulation. And interestingly, when you put those into a pathway analysis, you come up with the NF-kappa B complex as potentially regulating these genes. That's why we decided to focus on NF-kappa B as a potential target. So what is NF-kappa B? And I am almost done. NF-kappa B is a signaling pathway that eventually promotes survival and proliferation of lymphoid cells and some cancer cells. This is an example of one signaling pathway of NF-kappa B. The NF-kappa B is actually transcription factors that are in the nucleus that promote transcription of survival and proliferation genes, but they're held in the cytoplasm in the inactive state by the IKKs. The I-kappa Bs. So the I-kappa Bs bind and retain the transcription factor in the nucleus. Upon stimulation of the pathway by molecules such as TNF, you get phosphorylation of the I-kappa Bs and degradation in the proteasome, and then the transcription factors are free to translocate to the nucleus. So what we did was we actually looked at both IKK beta and IKK epsilon as regulators of this pathway to see if we could come up with a functional experiment to target that area in ovarian cancer. So we used the library in combination with either IKK high or IKK epsilon low, and we looked ovarian cancer cell lines, and we looked for the things that were specifically lost when IKK epsilon was low, in order to tie this together. Interestingly, we found CHEK1, which was highly synergistic with the loss of CHEK1 was highly synergistic with the loss of IKK epsilon, and vice versa. Expression of CHEK1 was very synergistic in promoting the survival of IKK epsilon high ovarian cancers. Interestingly, CHEK1 piqued our interest specifically in ovarian cancer because we found that it was overexpressed in Cancer Genome Atlas in almost 100% of ovarian cancers. So perhaps it has something to do with this whole pathway of genomic disarray. So what is CHEK? It works in regulating the cell cycle in response to DNA damage. So DNA damage occurs, and this is kind of a simplistic diagram, activates, in one sense, ATM, which then activates CHEK1 or CHEK2 to eventually halt the cell cycle at the G2M checkpoint. If you don't halt it there, the cell goes through its cell cycle, and then the damage kind of amplifies, and you get additional DNA damage, and the cell will die. So you can imagine that cells with a very messed up DNA damage response will have elevated CHEK1 or CHEK2 to block that response and allow the cell to sort of imperfectly repair the DNA enough so that they can then proliferate and go on through the cell cycle without going to apoptosis. So it makes sense in this type of cancer. We found that a CHEK inhibitor in our in vitro studies was most potent in high-grade serous ovarian cancer cell lines, much more so than in ovarian cancer cell lines representing other histologies. And we actually do have a clinical trial ongoing with a CHEK inhibitor that specifically that we have specifically using in high-grade serous ovarian cancers. We have a BRCA cohort and a non-BRCA cohort, and interestingly, we are seeing higher responses in people without a BRCA mutation, suggesting that this CHEK1 might be more important in women without BRCA mutations. This was highlighted, as I mentioned here, by a functional genomic approach instead of just sort of an observational screen. Okay, so in summary, this is kind of what I went through very quickly. Ovarian cancer is divided into epithelial and non-epithelial, which I didn't talk about. Epithelial cancers are high-grade serous, low-grade serous, mucinous, clear cell, and endometrioid. We have varying recurring abnormalities in the genome in each of these subsets, and we have focused on the homologous recombination defects in high-grade serous ovarian cancer with some moderate success. So just to tie it all up, in 1981, the discovery of HER2 was a functional genomic approach by throwing genes into mouse fibroblasts and seeing what worked. We've done shRNA library screen and thrown that in there and see what works when you knock it out, what kills the cell when you knock it out. So a functional genomic screen involves, ooh, that didn't come out good, the identification of a driver aberration or pathway, not just a potential marker. And then we've taken that to clinical trials with some success. And with that, I will stop and take any questions. Any questions? Yes.
This might sound like a weird question, but so homologous recombination is pretty important for like all cell types. And is there any reason you think that the BRCA1 mutations cause these cancers versus other types of cancers? Because you hear about them all the time in other areas.
Yeah. So the question was, why did the BRCA genes or loss of a BRCA function relate to ovarian and breast cancer and not other cancers, since all cell types need homologous recombination? And that is a fascinating question, and I've thought about that, for the last 10 years, because you would think that you would have any type of cancer, kind of like P53. I mean, leaf-Romney patients have multiple different types of cancers. So, I mean, one thought is that the breast and ovarian cells, because of the way they're cycling with a hormonal sort of influence, they might depend more on the BRCA-related homologous recombination as opposed to other cells. The other thought is that, I mean, these are all hypotheses. Nobody has a definitive answer. The other thought is that, yes, in leukemias, for example, when you lose, in blood cells, if you lose both BRCA genes, it's lethal. So the cell will lose the function of BRCA. It cannot divide, like it just dies. So like the cells in the blood, in the bone marrow might be getting, they're losing both copies of BRCA, but they can't form a cancer because they're just gonna like go away.
Yeah, so ovarian cancer, breast cancer might just be better off with no BRCA1 if they can find.
Exactly. So they might have the ability to survive. So it's kind of the other way around. Not that they're affected by the BRCA, but they are the only ones that can survive without it, because they have other ways of maybe upregulating CHEK1 and other DNA repair mechanisms like non-homologous end joining, for example, that can allow them to grow even though they have this loss of homologous recombination. Yes?
So why the ovarian cancer is diagnosed?
So the question is, why is ovarian cancer diagnosed in late stages, and what can we do? That has been a dilemma, for many, many years. Why it's diagnosed so late is because it does not present with obvious symptoms. And it's really hard to sort of examine that area. So for example, if you can think of breast cancer, the women can do a self-breast exam and notice that they have a mass in the breast. With ovarian cancer, the symptoms are, you can't really feel the mass until it's enormous. These symptoms are vague. They're sort of GI type symptoms: nausea, bloating, diarrhea, constipation. So the diagnosis goes for a while. I mean, it goes delayed because of those reasons, because there's very nonspecific symptoms. And then what can we do about it? People, of course, are looking for markers, but the markers that we found so far are markers of sort of peritoneal irritation, not specific to not specific to the cancer itself. So the marker, for example, CA-125 can be elevated for conditions like lung disease or pancreatic disease or any irritation of the bowel. Whereas so it's non-specific, but then it's also not very sensitive because there are 50% of cancers that don't have elevated CA-125. So we're looking for markers, but it is difficult. I mean, I think the big push now is for just awareness that pelvic and GI symptoms should look for a cause. The other reason is because the biopsy is kind of invasive, so it's hard to, the breast biopsy is a little bit easier to get to. Ovarian biopsies will be, more of a open surgical procedure. Yes?
For the same question, does the rapid progression of disease also play a role?
Yeah, so that's another interesting point that I was going to talk about. I didn't want to talk too long. That it spreads not. Oh, sorry. The question was, what about the rapid progression of disease? Does that contribute to the late stage? And I think that it's not necessarily the rapid progression of the disease, but it's the way it spreads. So a lot of cancers will spread through the blood and the lymph nodes. Ovarian cancer will, before it gets into the bloodstream, is going to shed. So it just sheds from the surface of the ovary. So it's very easy for it to sort of go and land on the liver, and go and land on the diaphragm. So it's very easy for it to float around and take up a new home. So I think that has to do with it as well. Yes?
Does any of the radiologic modalities play roles?
Yeah, so the question is, does any radiologic modality play a role in screening? There was just a recent study, the UKC TOX study, which looked at imaging studies, either CT ultrasound or transvaginal ultrasound, and in combination with CA-125 rise, so with a serum marker. And that did show some promise in screening for ovarian cancer, but it did not have as huge an impact as one would hope. The reason for that, again, is a lot of false positives with the screening detecting a mass that's not necessarily cancer, and so you have a lot of intervention going on for something that's not cancer. The early studies of that actually showed an increase in diagnosis, but it was increase in diagnosis of late-stage cancers and not early-stage cancers. So that's why they moved to sort of not looking at a particular cutoff of CA-125, but looking at the rate of rise of CA-125 marker. So, I mean, there's lots of ideas out there. But I think the take-home point is that the imaging has a lot of false positives, and so it's kind of hard to use as a screening test.
Thank you, Tina.
Okay, you're welcome.
Do you have any changes? Or?
Nope.
Okay. Okay, I have one more announcement, and that being I'll be sending all the registrants an email this week about the tumor boards and the core facilities. So this is all voluntary. If you'd like to learn more about what goes on at NCI, we have 3 tumor boards that you can visit: the medical oncology, urologic oncology, or the pediatric oncology tumor boards. And then we'll also have a tour of the pathology facility and the small molecule core, where they use robotics to develop new small molecules for cancer. So our next speaker is Stephanie Goff. She did a surgical residency at Columbia University. Subsequently, she went to the Brigham and Women's Hospital at Mass General. She's now in the surgery branch. She's going to talk about a hot new area, checkpoint modulation.
Yeah, I am. Thank you. So before I get started, how many of you are immunologists or in the immunology field? You might know more about T-cell activation than I do, but we'll get started. So to understand checkpoint modulation, you've got to kind of understand how the T-cell works and why we think cancer immunotherapy may be a good idea. So the objectives I'm going to try to go over today, we're going to talk a little bit about the mechanism of action of checkpoint blockade. I'm going to talk about the early clinical experiences we had here that I was actually able to participate in as a fellow when I was here before, and how we discovered what immune-related adverse events were and how it kind of changed how we had to think about how to run clinical trials using immune-mediated medicines. There are now 3 FDA-approved checkpoint modulators, and I'm going to go through some of the clinical data that led to those approvals, as well as then describe some of the more experimental uses of those same checkpoint modulators, as well as the development of a 4th that's not on this list. So just to know where we got started, had I made this slide 5 years ago, it would have only had 3 pillars, and those 3 pillars would have been surgery, which dates back to the Egyptians. They've actually found hieroglyphs that show them taking a breast cancer out of a patient. The next would be radiation, which got started in the late 1800s, early 1900s, kind of incidentally. And then chemotherapy, which got started at the Dana-Farber and then was more fine-tuned here at the NCI in the early 70s. Chemotherapy goes back to 1940. But in the last few years, particularly in 2012, and since then, immunotherapy has really become the 4th pillar of how to treat patients with cancer. We're going to go through a little bit of that today. So there are 3 main ways cancer immunotherapy can work. The first is nonspecific, stimulating the immune reaction of the body. We can either stimulate effector cells, and we've done this in the past using high doses of a hormone interleukin-2. We can inhibit the regulatory factors by turning off checkpoint blockade. We can actively immunize patients to get a more specific anti-tumor reaction, and that is a vast majority of the clinical data if you go out to read what's happening at places other than here. And we can passively transfer activated immune cells with anti-tumor activity, something called adoptive immunotherapy, which was really heralded here at the NCI by Dr. Rosenberg and continues to move forward as kind of a salvage therapy. What we're going to focus on today, though, obviously, is inhibiting regulatory factors or checkpoint blockade. So you need to know how a T cell gets activated. It's a 3-step process. It requires first signal 1, which is what provides the T cell with its specificity. When you have a protein, whether it is self or non-self, it gets eaten up by the proteasome in the cell, processed, and then placed on a major histocompatibility complex, which is really just kind of a pocket, makes its way out through the ER and back onto the surface of the cell. So any peptide that you might think could appear in the cytosol of a cell comes out as these small 9 to 10 peptide residues sitting in the pocket of an MHC. Now, our bodies have the ability to manipulate our genes to create a vast array of different T cell joining regions. And so you come up with specific T cells that can connect to this peptide in the pocket of this MHC. That's what's called signal 1 or T cell activation. It can happen with endogenous proteins. It can happen with endocytosed proteins, which generally get presented in the context of MHC2. All nucleated cells have MHC class 1, and they have this ability to reach in and pull out and put a small 9 to 10-mer in the pocket for the T cell to find. Professional antigen-presenting cells like dendritic cells have this ability to deal with peptides that get endocytosed, whether these are cancers, viruses, the flu you got last week. It brings it out and gives it to a CD4 or traditional helper cell, which then gives this specific T cell activation. But that's only the first step of the process. This T cell won't necessarily go and do anything until we get a little further. So the next step in T cell activation is that there has to be a co-ligand. So when the TCR next to the CD3, which is a marker of a lymphocyte, gets activated, it requires one of these second signals. Some of these second signals are positive signals that lead to all this downstream activation. But some of these signals, as you see pictured here in red, are actually negative signals. CTLA4, which is one form of checkpoint blockade, is one. If that gets activated, it will shut down a cell. PD-1 is another popular one. It will also shut down a cell. This second signal is what determines if this activated T cell becomes active or becomes anergic, stops working. It's just floating around doing nothing until it eventually apoptoses. The third signal, which is not as well elaborated as the other two, is the presence of cytokines in the area after a T cell has been activated. It can help polarize to one particular type of a T helper cell or T effector cell. And that is heavily dependent on the microenvironment in which it arrives. Signal 3 is something that's just now being worked out. These are things that would be more of the druggable target, small molecules. What we're going to talk about today is what happens at signal 2. So what is the role of our signal 2 checkpoints? Why do we have them? Why are they important? It seems like all they're doing is preventing us from killing cancer. What they're doing actually is protecting us from killing ourselves. So that's what prevents us from having autoimmune reactions to those T cells that find normal pieces of our body. So initial response to an antigen will occur in a secondary lymphoid organ. Now those can be the lymph nodes, which as you know we have all over our body, the tonsils, the spleen, Peyer's patches in the gut, mucosa-associated lymphoid tissue right under the surface of the stomach. And they allow us to recognize what is ourselves so we don't start to destroy it. The immune checkpoints also limit collateral damage. So say something gets activated and turned on, and then it goes to the site of, say, the splinter that you just got in your hand from running your hand down the stairwell. If you just let that T cell response go on, you could end up destroying your entire finger because once it turns on, it doesn't know how to turn off. So these immune checkpoints stop the collateral damage by shutting down that T cell response after it's gotten there and engaged. So in the context of cancer, we have 2 opportunities to break the tolerance to self-antigen. Because for the most part, cancers aren't something we get. It's not a virus. It's not something that our body can say, hey, that's not your DNA. Let's go attack it. It is your DNA. It is part of you, and we have to figure out how to trick your immune system into getting it. And this is one of the ways to do it. CTLA-4 is the first checkpoint blockade that was investigated by Dr. Allison, who just won the Lasker Award. I don't know if any of you are aware of that, becoming very prominent in the immunotherapy community. The reason is once this T cell gets engaged with signal 1, the TCR has engaged this peptide in the context of MHC. It gets turned on by activating CD28. Now, depending on the affinity of this complex, if this is a high binder, it will actually increase the amount of CTLA-4 that goes on the surface. And once that CTLA goes to the surface, it actually binds this ligand with more affinity than CD28. So even if you get something that turns on, the off signal comes on and it's stronger and it actually pulls it away. So then it turns the T cell off. So this is happening in the periphery when a T cell first encounters an antigen. It helps shut it down. It has a dampening effect so that once things get started, they don't get out of control. In mice that are double knockout for this, they don't die in utero, but they die very quickly of a lymphoproliferative autoimmune disorder within a few weeks. PD-1 is another ligand, excuse me, and this is more out once the cell that's been activated makes its way to where it wants to act. So you've had a T cell, it's engaged its peptide in the context of MHC, it's been turned on by CD28 or ICOS or one of the other positive stimulators, it gets to the tissue where it wants to be. It starts this inflammatory process. This inflammatory process makes the target cell express PD-1, which then shuts off the T cell. So it's a feedback loop at the site where the T cell is activating to shut it down. So any inflammation can upregulate PD-1, and it limits the collateral damage when you get an infection, when you get a splinter. It stops the immune system from going too far. So how does the PD-1, PD-L1 system work in cancer? There are some cancers, glioblastoma is one example, where the pathway within the cell actually just upregulates PD-L1 all on its own. It doesn't require a signal from the outside to turn it on. There's been a mutation within the glioblastoma cell itself that makes it upregulate PD-L1. But another way that we found is that when the system works, when we start killing the cancer, it releases interferon gamma. That interferon gamma actually upregulates PD-L1 and starts to shut the whole thing off again. So we get a little bit of success. The body says, hey, there's something going on here. We need to shut it down before it gets out of control. Unfortunately, we want it to get out of control because it needs more to kill the tumor. And so we got to find a way to try to block this response to interferon gamma. So with checkpoint blockade, where should we start? Where did we decide that we needed to get started? Well, we started with other tumors that were known to respond to immunotherapy. So the first is melanoma. There are about 10,000 deaths per year in the United States. Most of you, if you've ever had anyone in your family or anyone who's ever had a mole taken off, melanoma can be a scary thing, but the important thing to remember, 95% of all melanoma is cured by the dermatologist who does the first excision. The patients that need additional help, that develop metastatic disease, those are the ones that get into trouble. So once you develop metastatic disease, meaning it's gone elsewhere in your body, you have a 16% 5-year survival rate. Interleukin-2 was actually developed by Dr. Rosenberg across the street in Building 10 and can provide a durable cure. This isn't a word we use in cancer very frequently. These are patients that are alive 20 years later. 4% durable cure rate in patients who had widespread metastatic disease. The other cancer that's known to work with immune modulation is kidney cancer. So there's an estimated 15,000 deaths per year. Again, once you develop that widespread metastatic disease, your 5-year survival is only about 12%. And in these patients, again, done across the street at Building 10, durable cure in 7%. So we started with these. So this was the first in-human trial of anti-CTLA-4, which is the first checkpoint blockade. It was done, again, in Dr. Rosenberg's branch. Anti-CTLA-4, it's a monoclonal antibody that blocks that signal 2. It's called ipilimumab. It was a phase 1 trial. The patients got monoclonal antibody plus a peptide against GP100, which is a marker of melanoma. There were only 14 patients enrolled. There were 2 complete responders and 1 partial response. However, the accrual got stopped for toxicity. We had only treated 14 patients, and we started to see these really bad things. Dermatitis, where they would slough their skin. Colitis, bloody diarrhea, leading sometimes to perforation, requiring an operation. Hepatitis, causing fulminant hepatic failure. Hypophysitis, which is where there was a problem going on with the pituitary, and the patients would all of a sudden lack adrenal function, would lack testosterone, and would need to be put on steroid replacement. So while it had great effect, you see this is a lesion in the lung here, and you can see it's gone here. But what we were seeing, this is skin, and these dark blue dots, those are all neutrophils infiltrating the skin. On this side, this is a biopsy of colon. Again, all these small, round blue cells are neutrophils that should not be there. Lymphocytes that should not be there. And when you stain for CD3, which is a positive lymphocyte marker, that's what all these brown dots are. And this is the patient with hepatitis, and within, it's difficult to see at this magnification, but you've got lymphocytes kind of destroying the portal triad, so just destroying the bile ducts and the biliary system. So we stopped the trial. We had to figure out what was going on. However, this happened. After we stopped the trial, we kept watching these patients. 2 of these patients had every single bit of disease that they had disappear. So then we started to investigate, well, why is this happening? So we very cautiously started the trial again. We dropped the dose. We went down to 1 milligram per kilogram. What we found was if you had one of these autoimmune-related events, you actually had a higher likelihood of developing a response. In patients with melanoma, if they had a greater than grade 3 adverse event, you had a 36% chance of developing a response, whereas if you did not, you only had a 5% chance of developing a response. In renal cell cancer, the same thing. No patient that did not have a grade 3 AE developed a response. So then we decided, well, we lowered the dose. Now let's raise the dose. We think that causing these immune reactions, if we can get the patients through them, which we could, all of this could be reversed by steroids. If we can get them through the toxicity, if it cures them, are they willing to put up with that? And the answer was, of course, these patients are dying, and yes, they were willing to put up with whatever toxicity if we could rescue them from it. So then we started a dose escalation trial. What we found was that enterocolitis, again with bloody diarrhea that sometimes required an operation, was the most common grade 3/4 immune-related adverse event. And we did give steroids to manage these, and while we worried because steroids shut down the immune system temporarily, it actually didn't stop these patients from having a response. And what we found was that it didn't matter as long as you had any grade. So if you didn't have any immune-related adverse event at all, there was 1 patient that did get a response, but far greater numbers if you developed the immune-related adverse events. So what did we do here at the NCI for that? We kind of got that to the point where now it needed to go out into the world and needed to have a multi-institutional trial to get prepared. And once that happened, we kind of let it go and let them do the trial, let pharmacy do the trial. When we followed up our patients recently, we had an overall response rate of 13 to 20% and a 5-year survival of close to 23%. And more importantly, we developed algorithms for the management of these treatments that then went out into the world. So are any of you guys familiar with survival curves? Have you had enough clinical lectures to this point? So this tells you at the very beginning of the trial, all the patients were alive. At 12 months, 1 year, about half the patients were alive, depending on what trial they were on. Now that we're out here, what you have to keep in mind, though, we're at 7 years, and there are still patients alive. So these are patients with widespread metastatic disease who'd had one or more other treatments before they got to us. So they'd already used up part of that. Once you have metastatic disease, you have this long to live. They got to us, they'd already used up some of that, you have this long to live, and we've got them now living 7, 8, 9, 10 years. So that went out into trial, and it had a big national trial. It was published in the New England Journal. It had 11% response rate with complete responses in 3 patients. And you can see the survival curves here. Again, they didn't have as long a follow-up as we did, but at 4 years, they still have about 10 to 20% of patients alive. And so this was approved for metastatic melanoma, the first checkpoint modulator ever to be approved by the FDA for anything in March of 2011. Nivolumab was the next agent to be investigated, and this was against PD-1. So this was a phase 1 dose escalation, and in addition to the tumors that we knew could respond, melanoma and kidney cancer, this was a trial done at Johns Hopkins, and they took a number of different treatments: melanoma, non-small cell lung cancer, renal cell cancer, castration-resistant prostate cancer, and colorectal cancer. This is called a spider plot, and what this does is for each and every patient on the trial, it marks the length of the sum of all their tumors that we're measuring, and does it as a percentage. So at baseline, there's been no change. In patients who go negative, that means that their tumor has shrunk during the course of the treatment, and patients that go positive means their tumor has gone up. What they're attempting to show with this spider plot is that even though tumors don't go completely away, they stay away for a while. Now, keep in mind, this is in weeks, and so this is only about it's about 10 years here. This is a patient with non-small cell lung cancer. Something that we found out is when these immune cells rush in to attack the cancer, they actually make the cancer swell a little bit. So if this were a normal chemotherapy, when we went from this X-ray to this X-ray, on a normal chemotherapy, we would have said, that patient's progressed, it's time to switch therapy. Because we knew this might happen, we waited another 2 months, and now you can see those lesions are gone. So there's this pseudo-progression that happens as the immune cells influx into the tumor and then make the tumor go away. This was followed up with a bigger phase 3 randomized controlled trial where they tested, these are all patients, because ipilimumab was now an approved drug, the patients had to have already gotten ipilimumab, and ipilimumab had to have already failed to control their disease. So these are patients that are highly refractory to treatment. They got either nivolumab, which is anti-PD-1, versus chemotherapy of choice. This is called a waterfall plot, very similar to a spider plot, except instead of showing you, the course of an entire treatment like this does, it just shows you the maximum response. So any patient here that goes all the way down to 100, those are complete responders. Any patient that gets below this dotted line, that's a partial response. Partial response is defined as a 30% decrease in the sum of the longest diameters of the tumors. And then people are declared officially progressive disease if they go above 20%. On the basis of this, nivolumab was approved for refractory melanoma in December of 2014, so just last year. They then said, well, we used it in patients who had already gone through ipilimumab. How about if we try and see what happens if we use it first? So these are patients that hadn't gotten any kind of treatment yet for their metastatic disease. The reason they had to have wild-type BRAF is because another drug, which is not a checkpoint modulator, which is why I haven't talked about it, called vemurafenib, had been approved for metastatic melanoma because it works on BRAF-mutated tumors. In this trial, patients got either nivolumab or dacarbazine, which is DTIC, a form of chemotherapy. And you can see here, it's not often that you see survival curves split this nicely. So the yellow curve are the patients that got nivolumab, and the blue curve is the patients that got chemotherapy. It's a wide split. It was unexpected that we would see this kind of survival benefit. And at 1 year, almost 60% of the patients were alive in this versus 20% in this arm. What's interesting, though, is that this bottom graph, which is actually progression, shows that even if you progress, because this curve comes down a lot quicker than this one does, you still have a survival advantage because it's done something to keep your tumors under control. It hasn't made them go away completely, but it's keeping things under control. Pembrolizumab is just a different drug company's version of anti-PD-1. And it, again, was tested similarly to how nivolumab was in ipilimumab refractory patients to begin with. This was a phase 1 dose comparison. You guys know phase 1, phase 2, phase 3. Have they discussed that? So phase 1 is just looking for safety. Phase 2 is looking for efficacy. And then phase 3 is to show that it's better than what's currently available. So this was the same drug, just at 2 different levels. And it basically shows equivalence between the two. But more importantly, it still shows about 50% of patients alive at a year. So that's pretty good. So they said, you know what? This looks good. Keep going. Do a bigger trial. But we're going to go ahead and approve you for refractory melanoma. So they did the bigger trial. Again, dose comparison, but also versus chemo. And you can see here, the curves aren't as impressive as they were in the beginning, but still better than chemotherapy. So then they tried again, much like the other one, well, what happens if we give it first? So they tested pembrolizumab, the anti-PD-1, versus ipilimumab, the anti-CTLA-4, at 2 different dosing schedules versus ipilimumab. And you can see there's a difference in the response rates, 33% versus 11%. In a large number of patients. Again, the survival curves you can see here. Again, though, you saw immune-related adverse events, and it was higher in the ipilimumab arm. So with checkpoint modulation, the 2 approved antibodies interfere in 2 different ways. So anti-CTLA-4 blocks this interaction. We've got tumor on this side, T cell on this side. Anti-CTLA-4 works here, ipilimumab works here, nivolumab and pembrolizumab work here. Why don't we use them together? It's 2 different mechanisms. So they did that. So previously untreated patients, this is now a phase 3 randomized controlled trial, one versus the other versus the combination. What they did do for this trial, however, is you had to have PD-L1 on the surface of your tumor. So that's kind of selecting for a slightly better response rate than you might expect. It might be hard to see this, but the response rate in nivolumab alone, which is anti-PD-1, was 44%. The response rate in ipilimumab was 19%, and when you combine the 2, it was 58%. So over half of the patients had a shrinkage of their tumors by greater than 30%. However, the adverse events went way up in the combination. So you went from 16 to 20%, which is tolerable, to over half of the patients in the combination arm getting a serious side effect. This is just looking at those patients in a different trial who were defined as PD-L1 positive. You can see those patients seem to have a slightly higher response rate, but it's not a perfect biomarker because even these patients that are PD-L1 negative still enjoy a high response rate. So why melanoma? This is something we've been trying to figure out for a while. If you look at stuff coming out of the TCGA, melanoma has a very high level of non-synonymous mutations. So that's when you look at the tumor DNA, tumor RNA, tumor DNA, and you look at the normal DNA, you compare the two looking for a change in the DNA that leads to a change in the protein that's coded. So non-synonymous mutation. This is in a logarithmic scale, but you can see it ranges here. And I can tell you from the work that we've done across the street, we usually get 800 to 900 non-synonymous mutations per melanoma that we resect. So we figured, well, maybe because we're breaking self-tolerance, our immune systems already know how to find these mutations. These are mutations that came up in a patient's tumor. Their immune cells are already circling around them. Maybe these are T-cell responses to the mutations. Maybe that's what's doing it. So what are some other highly mutated tumors? Can we test those in this environment? So non-small cell lung cancer. You can see here we're no longer talking about rare diseases, 160,000 deaths a year from non-small cell lung cancer. Worse prognosis for patients with widespread disease. Only 2% are alive at 5-year survival. And a high correlation between smoking and the number of mutations. Another group of tumors that might be worthwhile looking at, tumors with mismatch repair deficiency. So these are tumors that either have a germline mutation in one of these 4 genes, plus or minus a couple others, that make it impossible for them to repair their mismatch during recombination. There is an additional group of patients that develop this sporadically, not in a germline. Lynch syndrome is a familial syndrome that passes down. And then a third type, bladder cancer, only about 16,000 deaths per year, but it is highly lethal once it's metastatic. So you've got melanoma, which is constantly exposed to UV exposure, which is why we think it's highly mutated. You've got smoking, which leads to mutations in the lungs. Bladder cancer, because every noxious thing that we eat or put into our body gets filtered through our kidneys and ends up in our bladder, so that's potentially causing mutations. So we decided to start testing these drugs in these diseases. So for non-small cell lung cancer, this is nivolumab. These patients have already gone through 2 other treatments. This is definitely third-line salvage kind of chemotherapy. They treated 117 patients and were able to get, a decent 14-15% response rate. And you can see in the waterfall plot here some pretty substantial shrinkages. And these patients in the blue are all alive. They did a randomized control trial versus chemotherapy, the same antibody. And you can see it's not as impressive as the other one, but the curves definitely split. And here at 1 year, you've got a 42% survival versus a 25% survival. So this was approved for refractory non-small cell lung cancer in March of this year. Pembrolizumab, again, just another form of the same antibody by a different company. 495 patients. Some of these patients had never gotten any treatment before. Those patients that were smokers, as you might imagine, because of higher mutation rates, had a higher response rate, 22%. Those that had never smoked, or at least reportedly had never smoked, had a 10% response rate. The adverse event profile for pembrolizumab is fairly small. However, one patient did develop an autoimmune pneumonitis that led to her death. This decision is actually going to be made in a couple weeks, so this might be yet another drug approved soon. So looking at those mismatch repair deficient cancers. So again, it's this hypothesis that the new antigens from the somatic mutations can lead to a T-cell repertoire that can be turned on by stopping the checkpoint blockade. So they use 3 parallel cohorts. These are all colorectal, these 2 are colorectal cancers. Those in a patient whose mismatch repair genes are proficient, those who are deficient, whether it's germline or inherited, and there are a few other cancers, ovarian, et cetera, that are also mismatch repair deficient. So focus on the colors for a second. Proficient is in the red. Deficient is in the blue and the black. You can see almost all the patients with proficient don't respond to checkpoint blockade. They don't have the same number of mutations, whereas all those in the blue and the black seem to be experiencing a decrease in their tumors. Again, more fuel to the hypothesis that this is all based on antigens, but again, we're building a lot of circumstantial evidence. No one's actually been able to elucidate the true mechanism of PD-1 and why this happens. Interestingly, though, when you look at where the tumor and the stroma interface, so this is traditional H&E staining of a colorectal cancer. The tumor is on the top left, my left, your right. Normal tissue is in this bottom corner. This is plain H&E staining, so you can see the tumorous glands here. These are disordered, kind of out of control, whereas this is nice, normal, smooth. This is staining for PD-L1. And almost all the reactivity is right on the edge of where the tumor hits the normal. This staining, CD8, again, all on the edge of where the tumor hits the normal. You can see here in a mismatch proficient colorectal cancer, almost no PD-L1 expression at all. What we do a lot is we take out these tumors and we look for the lymphocytes that grow in them. You can see the mismatch deficient colorectal cancers who purportedly would have more mutations have attracted more lymphocytes, whereas the ones that are proficient, don't have the mutations, don't attract the lymphocytes, have a smaller volume. So bladder cancers, again, very small numbers, only 29 patients, but at 12 months, 40% alive. It's fairly remarkable. This is not fully mature data. This was just presented in abstract form at ASCO. Tremelimumab is another form of anti-CTLA-4. It actually failed its clinical trial, so it did not get approved for melanoma. This is one of the few negative trials you'll ever see published. However, people are starting to use it in combination trials, although you won't find it in a lot because it failed to demonstrate a survival advantage. On a new front, we're starting to look at breast cancer. This is something that was previously thought not to respond to immunotherapy. But triple-negative breast cancer has a fairly high amount of PD-L1. 59 of the samples studied in this particular test had PD-L1. They treated these patients. They had a response rate of about 18%. There's a lot of controversy, though, on how frequently you should expect to find PD-L1 positive tumors in breast cancer, because another group coming out of MD Anderson found that PD-L1 was only positive on about 20% of triple-negative breast cancers. But you'll see this repeated over and over and over again. In about every different histology, somewhere between 15 and 20% of patients are able to respond to this therapy, which is remarkable. Gastric cancer, again, a very difficult disease to treat. Almost uniformly fatal. Again, 40% of them were positive for PD-L1. They had a response rate of, again, about 20%. Head and neck squamous cell cancer. Again, 25%, 20%. Now, interestingly, again with the theory that mutations can cause this, if you have HPV as the dominant driver for why you have a squamous cell cancer, you don't need a whole lot of other mutations to do it. If you don't have HPV, you need more mutations to do it, and you can see the response rate is slightly higher in those patients that are HPV negative than HPV positive. And again, circumstantial, but more evidence to put on the side that mutations are what we're reacting to. So this is the pipeline for checkpoints. These are all, well, not all the ones we know of, interacting between a T cell and an antigen-presenting cell or a tumor. Here's our main signal 1 TCR with its MHC class 1 or class 2. All these have yet to be investigated. CTLA-4 is here. PD-1, PD-L1 is here. Interestingly, PD-L1 has another receptor that we haven't identified yet. CD80, which normally acts as a co-receptor for CD28, can actually act as a ligand for CTLA-4. That's a new function for CD80. And so there are drugs coming out in the pipeline to try to break each and every one of these interactions. So anti-PD-L1, we've blocked it on one side. What happens if we block it on the other? Again, about 40%. They got a response rate of 36%. Bladder cancer, again, the waterfall plot that we've discussed before. The patients in green have high levels of PD-L1 on their surface, and they're the ones that are showing the best responses. So these are the drugs currently blocking the PD-1, PD-L1 pathway. Anti-PD-1, nivolumab. In melanoma, 32%. Non-small cell lung cancer, 17 to 30%. Renal cell cancer, 29 to 21%. Pembrolizumab, different company, same target, and you see the numbers there. Anti-PD-L1, none of these have been approved currently. These are the ones that are FDA approved. This one will be decided in a few weeks. This is kind of the current state of where we are with checkpoint blockade in the clinic. So again, checkpoint modulation. These are our targets, and this is where the next generation of immuno-oncologists are going to be working. So there's a whole lot of information I just kind of threw at you, but I'd be happy to take any questions. And or get you out of here early, whichever you'd like.
So I had a question in regards to T-cell development.
Mm-hmm. Mm-hmm.
So is the thought with these checkpoint inhibitors that you have an uniform T-cell receptor recognition to a antigen? And let's say that's a self-like antigen.
Mm-hmm. So our theory at the current time is that when in the course of your life you have encountered this antigen in the past, it has likely just been shut off. So it's become anergic. And that's through negative selection in the thymus. That's through a number of other mechanisms. But it's still out there. It's floating around. If you look at the genes that help develop a TCR, there's a joining region, there's a CD3 region, there's a TCR alpha, there's a TCR beta, and within those TCR alphas and betas, you have multiple different ways. You could literally, with the content of one person's DNA, make on the order of 10 to the 11th different TCRs. And so we have the capability of creating all those TCRs. If they don't encounter an antigen, that cell dies. So that T cell creates a TCR, it never encounters its antigen, it dies. The DNA gets read a different way, makes a different TCR, doesn't encounter its antigen, it dies. It keeps creating the cells keep creating TCRs until they find one they engage with, and then it either gets activated or becomes anergic. And then the thought is CTLA-4 works out in the periphery. It stops that initial anergic reaction, so it may lead something to apoptose that shouldn't have, whereas PD-L1 stops it from getting shut down at the second step. Does that make sense?
Yeah, that makes sense. I guess my question was after that.
Okay.
Looking at, so children, like they have this thymus present.
Right.
Versus look at older folks without thymus.
Uh-huh.
Thymus tissue present. I'm sure there are thymus independent T-cell
Mm-hmm.
development pathways. But you're talking about regeneration because the cells die eventually.
Right.
So let's say there was a antigen from a tumor. How would the body recognize that antigen if that cell, that T-cell has died off 10 years ago? Is it being regenerated?
It is being regenerated. So anytime a lymphocyte regenerates, it has the capability of making any TCR it wants. And so stochastically, it doesn't seem like it would ever connect more frequently than it does. But the way epigenetic modulation works, it tends to get in one certain family in one certain spot. It all depends on what your MHC is as to what you can recognize in the pocket of an MHC molecule. And so there's coordination between what TCR alpha and beta genes get turned on depending on what HLA you have. Because if you've got, say, HLA-A1, you're not going to start making TCRs that don't recognize HLA-A1. So we don't fully understand all the mechanisms, but there's enough there to crosstalk. Any other questions?
Is there any progress on checkpoint therapies for any of the tumors that are non-highly mutated, or like, for example, like epigenetically dysregulated tumors?
So a lot of that work has been pushed forth, particularly by Tom Gajewski's group in Chicago, trying to create a tumor that has a more inflamed look. So they joke about trying to make all the tumors look like melanoma. So what can you do to lead to an increased immune infiltrate in the tumor? Because if you can do that, then you can start to use these immune modulating things against it. So there's a trial in pancreas cancer at Johns Hopkins where they're trying to use a vaccine to create a more inflamed phenotype. And if they can do that, then they want to try to use some of the immune modulators against it to see if by creating an inflamed tumor, then they can turn down the inflammation and kill the tumor at the same time. So converting a non-inflamed tumor to an inflamed one.
And so you showed this slide with lymphocytes invading a tumor with mismatch repair.
Uh-huh.
Was that during treatment or after or before treatment?
This biopsy was done.
So this is with the drug.
This was with pembrolizumab.
Okay.
This was just came out in New England Journal just prior to AACR, I think, because
But that doesn't happen in the absence of that drug.
in their supplemental figures, you can actually see PD-L1 upregulated. This part of it happened after drug. This is a pre-treatment biopsy. Any other?
Thank Dr. Goff.
All right.